A very good afternoon. Welcome back to your online class. And today I am going to take your biology class from the chapter The Fundamental Unit of Life. No. And in previous class we have learned up to plasma membrane and cell membrane and how the osmosis and diffusion takes place. No. You have learned this all portion. Now today I am going to discuss about the three types of solution, how the movement of water takes place between outside and inside the cell. Means there is a condition what will happen if we put animal cell or plant cell into a solution of sugar or salt in water. Okay. So there are three conditions. First one is hypertonic. Understood? First one is hypertonic. Okay, again I am telling this all. Hypertonic, hypotonic, if the concentration of water outside the cell is higher than the concentration of water inside the cell, then the cell gains water by the process of osmosis. Okay, and water can move into the cell from the cell membrane, and in the case of hypotonic solution, more amount of water enters the cells and which results the swelling of the cells. No. Now the next is isotonic. Here what happens if the cells are put in an environment which has similar concentration of water as present inside. Okay, then this state allows for the free movement of water across the membrane without changing the concentration of solutes on either side. Got this point what I have told? So we can say that the size of cell does not vary in an isotonic solution because there is no net movement of water. Okay, as there is no net movement of water, so there is no change in the size of the cell. Yes, you can make one copy for science and make some note. Okay. Now isotonic we have finished. Now comes to hypertonic solution. If the cells are kept in an environment which has lower concentration of water than what is present inside the cells, and due to the process of osmosis water moves out of the cells, so this condition can be called as hypertonic solution, and this result in decrease in the size of the cell, or the resultant structure can be known as shrinkage. Okay, as more amount of water comes out of the cell. Okay, so these three solution you have already read, and again I have revised this portion. No. Now comes to the cell wall. No, you have to make different copies. Now first we will come to the portion that is page number 60. Give two examples of osmosis. Give two examples of osmosis. Can you write in your chat box what are two examples of osmosis? What are two examples? What are those examples of osmosis? Yes, very good, Sneha. Such a good example. Okay. Also there are some unicellular freshwater organisms which are or some plant cells that tends to gain water due to the process of osmosis, and also absorption of water of plant, that is also takes place by the process of osmosis. Okay, so these are, yes, transpiration. Very good. These all. Yes, Neha. So these are two examples. Now diffusion is the important in exchange of gases and water in the life of a cell, and in addition to this, cell also obtain a nutrition from the environment and different molecules that moves in and out of the cells through the type of transport requiring use of energy. So these are by active transport with expenditure of energy. What is the energy currency of our cell? What is the energy currency? Please tell me. What is the energy currency of our cell? ATP. What is the full form of ATP? Yes, adenosine triphosphate. What about T? Adenosine, not adenosine phosphate, adenosine triphosphate. Okay, triphosphate. So the plasma membrane is flexible and is made of organic molecules called lipids and proteins. So what are the main composition of plasma membrane? Lipids and proteins. Protein, lipids, proteins. PLP. Plasma membrane is composed of protein, lipid, protein. Okay. And however, we can observe the structure of plasma membrane only through electron microscope. Okay. There is an ultrastructure where the plasma membrane have lipid bilayer structure. Okay, means if this is a cell and if we magnify this portion only, cell membrane, then we are able to see the structure like the way, means if this is the plasma membrane, here lipid bilayer structure in both sides, such arrangements are there. Okay, means this is the head portion and this is the tail portion. This head is the hydrophilic head. Hydrophilic head. And this tail portion is hydrophobic tail. Now from the term you can conclude what is this. Hydro means water, philic means love, means that loves water or that attracts water, that is the hydrophobic head portion. Okay, and hydrophilic head and hydrophobic tail. Tail means here hydrophobic, phobia means fear, that is, or hydro means water, that is away from water. So the tail portion that is away from water. So inside both lipid bilayer structure, you can see here the middle portion, these are the tails, and tails are hydrophobic in nature and the heads are hydrophilic in nature. Okay, yes, and there are oceans of lipids which are in which proteins are embedded according to the fluid mosaic model. Okay, now you understood what I have told? Okay. Now comes to one process that is known as endocytosis. What is endocytosis? It is a process by which plasma membrane that engulfs or captures food or other materials inside the cell. Okay. So what is endocytosis? The process by which the plasma membrane engulfs food or other materials inside the cell. This is known as endocytosis. Okay. Everyone understood? Okay. Now let's proceed. So we have seen up to endocytosis. Now comes to the portion that is cell wall, and we know that cell wall is the outer and hard covering of cell and which maintains the shape of the cell. Okay, and the cell walls is generally made of cellulose. Now can we digest cellulose? What do you think? What do you think, that we can digest cellulose or not? Yes, Jasmine. Plasma membrane is called permeable membrane, that limits entry and exit. Some of the means it permits only. Okay. Yes, ruminating animal can, but why we can't digest cellulose? Give me the reason. What is the main reason behind this? We know cellulose have to broken down, but why we can't digest cellulose? What is the main reason here? Yes, it is too complex for us, but another reason is there. Another prominent reason is already there, Jasmine. You have to mention that. Now I am telling, we lack cellulose digesting enzyme in our body. That's why we can't digest cellulose. Okay, we lack enzyme this cellulose digesting enzyme. Okay, that will be the answer. I hope you understood. Now proceeds plasmolysis. As you have learned in hypertonic solution, plasmolysis is the process in which the contents of the cell that are away from the cell wall shrink or contract when a cell loses water due to osmosis or when it is kept in hypertonic solution. So the case plasmolysis that will occur in hypertonic solution. Now you can easily differentiate between hypertonic and no. I have just told you plasmolysis occurs in hypertonic condition. Okay, plasmolysis, that is the event. Okay, yes Jasmine, that will be the answer. Your answer is correct. Now just answer me, can dead cell absorb water? Can dead cell absorb water? No. Yes, very good. Dead cell cannot absorb water through osmosis. Yes. Now, how plants, fungi and bacteria can exist in hypotonic medium? Actually, plants, fungi and bacteria, they exist in such situation because of their rigid cell membrane. As you know, rigid cell membrane, plants, fungi, bacteria. What is their rigid cell membrane? Plants, rigid cell membrane, cell wall. Yes. So, I have told you, even if the cell swells up, the cell membrane is able to prevent them from bursting out. So they have such a rigid structure. Okay, I hope you understood. And Jasmine, you have a question in previous class, I think, about blood. Please ask me now what you have asked for in previous class. I think you have asked for blood grouping, no? How antigens or antibodies are present in different blood group. Such type of question. Oh, okay. Classification. Okay, actually there is ABO blood grouping system is present, and in addition to this ABO blood group, there is an prominent blood grouping that is known as RH factor or RH blood group. Okay, now I will tell one thing, that as you know what is plasma. Do you have any idea about plasma? In blood, 55% is plasma. Okay, 55% is plasma, and 45% is RBC. 45% is RBC. You know this? Now the WBC means white blood cell and platelets, they are embedded on the plasma. Okay, so total 55% blood is plasma and 45% is RBC, and another WBC and platelets, they are embedded on this plasma. Okay. Now there are I have told you ABO blood group, they have 4 groups. Okay, they have 4 groups based on the antigen and antibody present in them. The ABO blood group can be grouped as 4 groups. Okay, A, B, AB and O. Okay, these 4 blood groups are present. Yes, Jasmine, liquid substance, this plasma I have told you, that is actually contains 92% of water and 7% vital proteins are there, just like albumins. Okay, and 1% it consists of salt or hormones, sugar, etc. Okay, now I will classify this blood group into 4 categories. In first categories, you have to remember one thing, as this classification is on the basis of the antigen and antibody present. Actually, antigens you know, these are foreign particles, and antibody you know, these are protein in nature or immunoglobulin. Okay, and one thing, antigen, these are actually antigens, these are actually found in the surface of RBC, and antibody, these are embedded on the plasma. Okay, now when in case of A blood group, what happens, there is antigen A is present and antibody B is present. Okay, so here antigen A is present and antibody B is present. Okay, in case of A blood group. And in case of B blood group, just opposite of it, antigen B is present, and antibody A is present. And in case of AB blood group, there is no antibody of A and B, only 2 antigens are present. Here antigen A and antigen B are present. Okay, in case of AB blood group. And in case of O, just opposite of AB blood group, that is there is no any antigens present, only 2 antibodies, that is antibody A and antibody B are present. So here no antigen is present in case of O blood group, and in case of AB blood group there is no antibody are present. So that as there is no antibody are present, AB blood group is known as universal recipient. Universal recipient. Okay, universal recipient. And O blood group is known as universal donor. Okay, understood, Jasmine? That will be actually based on some WBC or platelets. Okay, as you know, WBC, what is the main function of WBC? Okay, Jasmine? As you know, there are so many types of WBCs are also there: neutrophil, basophils, eosinophils, monocyte, lymphocytes. So on the basis of their numbers, we will able to see that the person who are more prone to infection, okay, and who are less prone to infection. Okay, I hope you understood. Okay, now let's comes to the next portion of your book, that is nucleus. As you know, nucleus is the prominent organelle that is present in cell, which is the controlling center of all activities of the cell. Okay, that is the controlling center of the all activities of the cell. Okay, and from the structure of nucleus we will able to see that the nucleus has a nuclear membrane which covers its all around, and there are some pores which are present on the nuclear membrane that allow the movements of substance and in and out of the nucleus. Okay, so such nuclear membranes have pores so that that allow the movement of substance in and out of the nucleus. Okay, understood what I have told now? Okay, so now comes to, there are chromosomes, as you know, chromatin fibers, these are present inside the nucleus, and they are rod shaped structure present in the nucleus which contain genetic information. What is that portion known as? Genetic information, that carry genetic information. DNA, yes, deoxyribonucleic acid. What do you think that what has evolved first, DNA or RNA? Tell me. Among DNA and RNA, which has evolved first? Which has evolved first? Yes, very good. RNA. Correct answer. Now I have told you there are chromosomes. They are rod-shaped structure present in the nucleus. This chromosome actually contains two types of things. Okay, chromosomes are mainly made of two types of things. One is DNA and another is protein. Okay, one is DNA, another is protein. Chromosome contains these two types of things. One is DNA, another is protein. Now what is the main function of DNA? DNA is the mainly responsible for organizing and constructing new cells. So what is the main organization center? That is DNA, where the construction and organization of the cell takes place. Okay, in DNA. And proteins are actually helps in packaging and condensation of DNA. So the DNA can be compacted in a condensed way. Okay, and as you know, the genetic information that is transferred from one generation to the next generation. Okay, and chromatin, another thing we have come across. Chromosome now comes to chromatin. Chromatins are thread-like material that are present in the cell, and chromatins organizes itself into chromosome whenever the cell is about to divide. Okay. I hope you understood. And in the next class, further we will discuss the more detail about this chromosome. Okay, chromosome, then chromatin, these all DNA. I hope you understood, and if you have any problem, you can ask me now.